Estimating electric signal preference in Paramormyrops kingsleyae. Weakly electric fish are able to communicate and interact with their environment via low-voltage electric organ discharges, or EODs. The species we studied in this experiment, Paramormyrops kingsleyae, exhibits remarkable variation in its signal. Some populations have two peaked biphasic EODs, while others have three peaked triphasic EODs. Previous studies have shown that P. kingsleyae individuals can distinguish between both EOD types. Our study aimed to determine whether they exhibited any preference for biphasic or triphasic EODs. The experiment was conducted in the summer of 2019 during the Gallant Laboratories trip to Gabon. Fish were tested from 4 different rivers: Apasa in Bambomo in La Bamba, and Ebunji and Mengono in Cocoa Beach. Mengono and Bambomo kingsleyae have biphasic signals, while Apasa and Ebunji kingsleyae have triphasic signals. One fish was designated the test fish and was placed in a tank that was connected to a stimulus fish in a separate tank via electrodes. As the stimulus fish sent EODs, the signals were first processed through a computer. The computer acted as a voice changer. It played a biphasic waveform from one electrode in the test fish tank and a triphasic waveform from the other. EODs from the test fish were sent back to the stim fish, allowing the fish to communicate in real time. Each test fish received 2 different sets of biphasic and triphasic waveform signals, and each set was played twice to control for side bias. Signals were played for 2 minutes each trial. The second part of the study involved analyzing the data from the trip. We used Boris, a video logging software, to analyze several behaviors. During analysis, the observer was unaware of which signals the fish was receiving. Each video was watched by 2 different observers to minimize observer bias. We analyzed the amount of time spent near each electrode, as well as 3 different behaviors: circling, headbutting, and tail probing.
For the behavior, circling was defined as the fish inspecting the electrode while completing at least a 3/4 circle around it. For headbutting occurred when the fish would attack or lunge at the electrode. For tail probing is when the fish would quickly wave its tail where the fish's electric organ is housed near the electrode. As a side note, most fish did not perform headbutting or tail probing behaviors regularly. In terms of differentiation between time spent between the 2 stimuli, we can see that there isn't a sizable difference between their preference of waveforms when taking into account standard error, as they for the most part overlap a sizable amount. The only group that somewhat breaks this trend is the fish from the Apasa River, as there is the most noticeable difference between their preference between the BP and TP waveforms. This group seems to slightly prefer the BP waveform in this case. In terms of the circling data, the fish had a far more varied spread in comparison to the time spent near the electrodes. Although in some cases the average number of circles is greater for one signal than the other, across most rivers the standard error of each of these points mitigates the drastic differences between the circling of the 2 electrodes. Bambono is the only one that has the highest difference between the amount of circling between the BP and TP signal, even when taking into account the standard error.
In order to statistically evaluate the difference in preference, we generated strength of preference values by looking at the conspecific and heterospecific stimuli, where the conspecific refers to the same EOD waveform and heterospecific refers to 2 different waveforms. We calculated these for the amount of time spent near the electrodes by taking the time spent around the conspecific playback stimulus, subtracting the amount of time near the heterospecific playback stimulus, and dividing that by the sum of the total amount of time spent near both the conspecific and heterospecific playback stimuli. The strength of preference values can range from -1, indicating complete avoidance of the conspecific stimulus, to +1, indicating complete preference for the conspecific stimuli. The SOP values were then tested against a random distribution centered at 0 using a 1-sample t-test, and yielded the t-statistics and p-values that are shown in this chart. All of the p-values for time spent are greater than 0.05, which indicates that there is no significant evidence to indicate a preference for either EOD waveform. The same strength of preference values and 1-sample t-test were also calculated for circling behavior, which yielded the t-statistics and p-values shown in this chart. The p-values for the circling behavior are also all greater than 0.05, which indicates that there's no significant difference in preference between the triphasic or biphasic waveforms in any of the 4 rivers. Since our data does not show a strong correlation in signal preference, one confounding variable that may have affected the results is the sex of the fish. There have been other studies that suggest the presence of signal preference in males but not females. For our experiment, more analysis is needed in order to hypothesize if the sexes of the fish impacted the way they interacted with the electrode signal. Another confounding variable is the age of the fish. Some individuals may not have reached mating maturity yet. It's also possible that a smaller, younger fish hearing the EOD of a larger, more aggressive male may behave more timidly, and its behavior would likely not match the expected mating behavior. Another variable is novel or unfamiliar EODs. The fish used in the experiment come from 4 different rivers, some of which are completely isolated from each other. It's very possible that fish with biphasic EODs have never heard a triphasic EOD, and vice versa. This suggests the possibility of certain fish interacting more with a particular electrode, not because of mating preference, but because of its curiosity about a novel EOD. In conclusion, so far there's no major observable preference in electric signal waveforms. However, we are currently doing more analysis on the amount of time each fish spent directly interacting with the electrodes, and in the fall we're hoping to analyze the stomach contents of the fish. This could help us investigate other possible factors, such as diet preference, evading predators, or random genetic drift, that could be contributing to the diversification of EODs in these weakly electric fish.